And then the main thing that we have been focusing on is essentially looking at temporal, magnetic field microscopy. And so I'll be telling you about the dynamic wide field image technique that we have developed, and we have a patent for this, and highlights of some other work that we've been doing with NV centers in diamond. So just to touch upon, because this is a large audience, and you have been probably hearing a lot about different kind of quantum systems. So we work with NV centers, which are extremely good single photon sources as well. It's not just NV centers, but there are other kind of I'll tell you what the details of the NV centers are, but there are other kind of defects like silicon vacancies, germanium vacancies, etc., which work as very nice single photon sources. And so here, if you see on I hope you can see my mouse. So you see here is an image of the different kind of the photon sources that you can see from here, which is in an array. This is from NV centers, whereas if you have many NV centers inside the diamond, you can see that the diamond can really glow absolutely bright orange as shown in this figure. So essentially all the fluorescence that is emitted by the qubits are all now captured inside the diamond lattice, because it's a very high refractive index material, and so the light basically stays very nicely inside. In fact, it is very difficult to extract the light out, single photons out from the diamond, and it is one of the challenges. And therefore, people start making these kind of photonic structures, for instance, solid immersion lenses, as you can see here, where essentially, if you see, this is basically a little dome-like structure, which is built around a single photon source. So the source is actually in the center, and you can build a photonic structure so that it enables you to outcouple the light very highly. So here we work on silicon, silicon photon sources, silicon vacancies in diamond, which are single photon sources at 742 nanometer. And you can see here the sources are right in the center of these photonic crystal structures. And what we intend to do, and we have been working on, is essentially trying to couple the spins and the photons together, and trying to understand how one can actually sorry, I don't know what happened. So one can, couple the spin and the photon and understand the interactions of the spins and the photons, and understand the photonic coupling with spins. And this is one of the key also applications of one of the things is development of memories. And essentially, you should be able to interact with the spin, which is, your qubit, and then the photons, which can carry the information and then become like a node where you can have a memory unit, as well as you can carry the information again. So this is like a sort of a quantum repeater node as well. So there are various different applications. We have only been focusing on some of them. Now I'm going to jump into magnetic field sensing and magnetometry. And here, what's happening? So here you can see this is a plot of sensitivity versus linear dimensions. And in this plot, essentially what you see is that there are different kind of sensors that are available. So you have all sorts of sensors, for instance, QIT sensors, NV sensors, optomechanical, chip scale electronic. There are various different, magnetic field sensors, and they all cover different ranges. For instance, you can have very sensitive sensors like the atomic vapor sensors, which can sense magnetic field in the order of femtoteslas. And femtoteslas, just to give you a sort of understanding of scale, femtoteslas is essentially means that it's 10 to the power of minus 15 Tesla. So Earth's magnetic field is of the order of 50 microtesla, and this is like several orders of magnitude lower. So you have extremely high precision magnetic field sensors. You also have NV centers, which fall in the range somewhere between the best reported, for instance, are in the order of picoteslas. But the advantage of NV centers is that because it's a sample which can have a very uniform NV center base, you can use it for imaging microscopic magnetic fields. And there really NV centers play a key role in magnetic field sensing, where you can actually map out the magnetic field that is near the NV center. So it can be used like a imager, unlike atomic vapor magnetometers, where it's like a bulk sensor, while you get very high sensitivity, but you can't really use it for imaging or any of that. So there are different applications that one can use for all these different kind of sensors. And of course, there's a lot of push towards developing portable sensors with NV centers, with atomic vapor sensors, and they have various different applications, for instance, in oil and gas sensing, for anomaly sensing, and lots of different kind of applications which are. And so these sensors, magnetic field sensors, are very, very important, not just for commercial purposes, but also for fundamental physics, understanding magnetism at the fundamental scale, where you can image, for instance, electron flow, or you can also understand phase changes, and these kind of things. Information can be extracted using NV centers as imagers, for instance. Now, there are other two applications that I would like to point out here, where NV centers are very useful is, for instance, in NMR, local NMR or nano MRI. So you might have heard of the term nano MRI, and that's essentially what it's doing is that you have an NV center, as you can see here. This is a spin, and if you have a protein molecule next to it, then you can actually understand the dipole-dipole coupling between the nucleus spins of the protein and the nucleus spin in the NV center itself. And so therefore, what one can do is, from all these dipole-dipole couplings, one can actually understand the protein structure and try to evaluate the protein structure. Now, why is this important? We already have methods like X-ray diffraction and all sorts of extremely sophisticated methods for doing protein structure determination. But one of the main challenges there is that the proteins have to be crystallized. Now, 99% of the proteins in our body and in mammalian cells, etc., are not crystallizable, and therefore techniques that allow you to form and understand the the crystal structure for developing new drugs, for developing understanding different kind of molecular interactions, you actually have to understand the protein structure, which proteins that cannot be crystallized. And therefore NV centers can really provide you one such solution where you can really understand the crystal structure. And I've also mentioned that NV centers can also be formed like a scanning tip, like an example of an AFM can be converted into a scanning tip NV center magnetometer. And so not just imaging, but you can also scan and understand different kind of magnetic behavior in various different samples, especially like superconductors, and high TC superconductivity is one of the areas in which NV center magnetometry can be really applied, trying to understand the fundamental dynamics of what the the sample is doing in the presence of defects or different lattice structures, etc. Now I want to move on to really understanding and telling you what these diamond NV centers are. So I'm sure many of you would have seen colored gemstones, and colored gemstones are nothing but actually defective crystals. What essentially happens is that when the diamond is being developed inside, let's take the case of a natural diamond, which is essentially is found in the Earth's crust, and has been formed because of extremely high pressure and high temperature, where the carbon really coalesces together and forms the diamond lattice. Now, as we know that nitrogen is highly prevalent in Earth's atmosphere, so it's almost 78% or so nitrogen is there everywhere. And so when the diamond is actually grown, you can have nitrogen that can actually seep into the lattice structure of diamond, and so that gives it a bright yellow color. You similarly can have orange color depending on the concentration of the nitrogen. And then if you have boron, which is also there in the Earth's crust, it can seep in and it gives you this bright blue kind of color. Now in the lab, of course, you can grow diamond, and this has become a very nice technology, which is CVD or HPHT methods of developing diamond. You can see that they will be essentially looking like glass plates. These are sort of CVD grown plates, and the plates can then be either crushed into forming what is known as nano diamonds, and then you can also use the plates, for instance, to grow some or develop lithographically develop pillars, or you can make, for instance, these kind of pillars which you can attach to an AFM and form the scanning tip, etc. Now, of course, in the lab, none of the diamonds look as bright and shiny as like the gemstone ones. What you get are something looking like colored glass, which is yellow, what you can see here. In my lab, we have these yellow and blue diamonds, etc. And the standard, very standard looking diamond samples that we have will actually look somewhat like this, because, you know, you really cannot distinguish between glass and diamond unless you know that it is a diamond and you perform some X-ray diffraction, or you can perform XPS, or, you know, or ESR and all these methods. Now, a very special property of these diamond samples is that when you have a nitrogen that has been substituted in place of carbon, and you also have a vacancy next to it, along with an electron that it captures from the environment, it forms what is known as the NV- complex. And this is the most interesting complex that is useful for forming the quantum sensors. And it has very interesting electronic structure and properties, which were accidentally found in almost around the 1950s. And people knew of various, there are several defects, not just nitrogen, but various kind of defects in diamond, but nitrogen vacancies seem to have a very special place in terms of the electronic structure. But of course, now, given that research has progressed a lot in diamond and diamond-based quantum technologies, people are looking into really understanding electronic structures of germanium, of tin, and are understanding that there are very interesting properties as well, which are comparable to NV centers and can be useful for various different applications. So NV- centers actually give the diamond a pink color. And if you would have heard of the pink star diamond, that's essentially one that has very high concentration of NV-. Now, this is very difficult to typically get. So any sample which we buy are typically from Element 6, which is a company that you have from which is a separate branch of De Beers, which is the, I'm sure you would have heard of this, the diamond company. Element 6 has developed several procedures of understanding how to really reliably create these NV- centers. And NV- centers, not just in bulk, but in a layer, understanding the different growth strategies of making these kind of samples is very critical for making a quantum technology that will be useful, scalable, and useful for various different applications. So you don't want to rely on naturally growing diamond or NV centers. You actually want to be able to get NV centers which can be grown in the lab so that you can make commercial devices. Now, going into the electronic structure here, you can see that this electronic structure is actually embedded in the valence, so in the band gap of diamond, which is a very large band gap, 5.5 eV. So here you have the NV- electronic structure. So it's a ground state triplet. It's also an electronic excited state triplet. Now, the interesting part about NV centers is that it is a room temperature qubit. It can be excited with a green laser, which is off resonate. So you can excite the green, the NV center from its ground state to the excited state. And then as it, decays down from the excited state, the zero phonon line is actually at 637 nanometers. And that's red color. Okay, and so essentially, if you have a very large range of emission, so it has a fairly large range of emission because of the phonon bands here, it can go all the way from 630 nanometers to almost 750 nanometers and even up to 800 nanometers in the range, and that's almost red color. Right. And so, but this is the zero phonon line, and that's the single photon emission band is actually 637 nanometer and 2 nanometers around it. Now, NV centers, the interesting part is that you can actually control the spin qubits, and the spin information is encoded in the optical information. And that's because when you excite the green with a green laser and it emits the red light, there is a possibility always that there is the NV centers can go to a metastable state and also decay back down to this, zero state. And And whenever it does that, whenever it takes this path, it always fluoresces 30% less, or at the most 30% less. And so if it were to start from plus or minus 1 level, the fluorescence is actually lesser in comparison to this 0 state. And that gives us a fluorescence contrast that allows us to distinguish between the 0 state and the plus minus 1 levels. And I'll tell you how this looks. So let's start with just the excitation. If you excite with green laser, you get a red fluorescence. In bulk, it looks like this. If you actually zoom in to a sample that has specific single point defects, like the single NV centers here, you can see that this will actually look like these bright patches. So the red is higher fluorescence and blue is nothing. So essentially, you have NV centers that are in patches over here. And if you zoom in, we have built a confocal microscope. You can do this measurement with a confocal microscope. You can land up with a point defect like this inside the diamond lattice. Now, of course, this is a very non-uniform distribution, and unlike the one that I showed you previously, where it was actually implanted with a targeted implantation. Here it's a non-uniform distribution. It's just a direct implantation without any patterns or anything. And so you can see that there is various different NV centers all over. This happens to be a single NV center, as we know from other measurements that we have performed on the NV center. Now, a qubit, just to, this is a spin qubit. As you can see, 0, -1 or 0, +1 are the levels that are used as the qubit levels. So you can, it's like a 3-level system, but because you have 2 distinct levels in the presence of a magnetic field, you can apply the microwave radiation and control the state from 0 to -1, and that can be your 2-level system. And you can also go from 0 to +1, and that could be another 2-level system that you can control. Now, -1 to +1 is a forbidden transition. And so on the block sphere, 0 and 1 state, and you can apply different pi pulses depending on this microwave, and you can do all the qubit operations on the NV center. Now, let's try and understand how the NV center works like a magnetic field sensor. So as I have already mentioned, the 0 state has high fluorescence in comparison to the plus or the -1 state. And so when we start our measurements, let's say on the fluorescence axis, we measure some number of counts, fluorescence or counts per second, right? Now, let's start applying the microwave. And as we apply the microwave, this is essentially an electron spin resonance. So whenever it hits the +1 level or the -1 level, you essentially will see a drop in the fluorescence. And so it's essentially whenever it crosses the resonant frequency, you will see that the fluorescence is minimum. And so what you plot is this blue curve here. As you trace out, as you sweep the microwave frequency, you will trace out a resonance curve that looks like this blue curve. Now, when you apply a biased magnetic field, what happens is that these lines, the plus and the -1 levels, will Zeeman shift with respect to each other. It's not just BZ field. You can also apply, any arbitrary field. This can shift with respect to each other, and therefore, this you can measure because this is an electron spin. Remember, so it's a very well calibrated system. Nature has given us the calibration of the magnetic field. It's gamma is equal to 2.8 megahertz per Gauss. So every time, whatever frequency shift that you observe, you can calculate exactly the magnetic field corresponding to that frequency shift. So that's how we find out what the magnetic field shifts are. And this is one of the 0th order mechanisms of finding out what the magnetic field measurement is. You can also use Ramsey, or there are other higher order techniques for measuring magnetic field. Now, NV centers, like I said, are not just magnetic field sensors. You can have electric field sensing, you can have temperature sensing, you can also have stress sensing, etc. But another interesting property of NV centers is that, remember, this is a diamond lattice, and therefore the nitrogen can be on any of these 4 locations, as you can see with the yellow, the green, blue, and the red location. And so the vacancies next to each other actually mark out different angles of the orientation of the NV center with respect to the diamond lattice. So it's along the 111 axis of the NV center. Okay, so that allows us to actually measure different projections. So if you have a field that is in an arbitrary direction with respect to your sample, you essentially will measure the projection of the field on the NV center axis, right? And so what you see here is, instead of seeing one Lorentzian which corresponds to the resonance, you see 4 on either sides of the 2.87 gigahertz, which is the zero field splitting. So this is the zero field splitting. And so you will see 4 lines corresponding each to the -1 and +1, depending on the NV center axis and the crystal orientation, right? And so based on this, this is a very important, aspect of NV centers, because now you have a lot of information about the projections, right? And from the projections, and because you know the diamond lattice very well, we can actually reconstruct back the exact magnitude as well as the direction of a magnetic field. So it's a vector magnetic field sensor. So and also you can see that the temperature and magnetic field and electric field all have different kind of interactions with the resonance, and so one can reconstruct back the temperature as well as magnetic field, or at least 2 of them simultaneously, has been reported in literature. And if you're interested, there's a very nice there are several review articles, but 2 of them I would point out here, which are very nice, and you can go into it for understanding magnetic field sensing with NV centers. Okay, let's understand how NV centers can be used as an imager. Now, you can have a whole bunch of NV centers in a sample which is very close on the surface of the diamond, and then you can have some magnetic sample. It could be anything like a 2D material or a 2D, let's say, ferromagnetic material that can have different, profiles of magnetic field. And what you would like to do is essentially sense the magnetic field that is close to it, because if all the magnetic field will do is actually perturb the electronic structure, level structure of the NV center, and that is encoded in the optical information. So it's called optically detected magnetic resonance. And from the magnetic resonance, you can essentially, from the red light, if you impinge that on a CCD, it has a spatial profile. So it can be a spatial imager, and that's how we built a quantum diamond microscope, essentially. So to give some of the numbers, DC sensitivity is of the order of 50 picotesla, 0.9 if you have flux ferrite. These are some special concentrators. And then if you can see that AC sensitivity, so NV centers can not only detect static magnetic fields, you can also do that dynamic magnetic fields. So AC sensitivity, and then you have 250 nanometers of spatial, because this is, diffraction limited ultimately because of the optics that we use. And then you have also, if you have a scanning probe magnetometer, then it is about 5 nanometers of spatial resolution. Field of view is very large because you can do a full wide area illumination and collect the photons and be able to image very large areas of magnetic field profile. So this is a very interesting application, and this is what we have been focusing on in terms of developing a quantum sensor. Now, just to highlight what is the aspect of quantumness that we are using really when we are talking about NV centers as quantum sensors, right? Now, the minimum field that we can detect depends essentially inversely on the T2, which is the coherence time of the NV centers. So coherence time is essentially means the fidelity, for how long can you measure the spin and be able to tell whether it is 0 or 1. So that's essentially in a coarse way of saying this is the fidelity with which you can do a measurement. So that is the T2 coherence time of the NV center. And then you also have the number of sensors. Now, classically, you know that if you perform a measurement n number of times, you get better sensitivity, or you get better resolution. And so essentially it's the same thing. If you have, instead of one NV center, you have many NV centers, you scale as 1 over square root n. And so that gives us the minimum sensitivity of the making this a quantum sensor, because here in the first order we are using a qubit with a certain coherence time, and with a certain number which scales as square root of n, you can get the sensitivity. Now, the interesting part of NV centers is that one can actually use real quantum mechanical, enhancement techniques. For instance, one can use entanglement within the spins itself if you have some sort of interaction between the spins, or you can engineer the interaction between the spins by somehow interacting it with a cavity, or really be able to create these special entangled states. Then you can actually go as the sensitivity will go as 1/n, and that is the real power of a quantum sensor, because you can approach the Heisenberg limit with engineered systems of quantum sensing. Now we work in this domain. We are not yet approaching this 1/n number, although we are developing strategies for this kind of technique. Now, to give you some examples, because this is a wide audience, there are different kind of applications that people have already been working on. For instance, wide area imaging of vortices, and that's very interesting because you can understand interactions of dynamics of electrons, like I mentioned, and then you also have vortices. And one other very interesting application, and where I feel that NV centers are really powerful, is because of the vector nature of the magnetic field. You can actually image not just the magnetic field, but if you have a current flowing wire, you can reconstruct back its J. And this is an application where they have actually looked at a graphene sample, and then they have flown the current and try to understand the hydrodynamic flow of current in graphene. And there are also other 2D materials also, where people are looking at different kind of devices and are trying to understand what is the current flow inside these samples. And these are some very nice applications. Now, another very contrasting application set is the biological domain, because diamond is a highly biocompatible substance, and therefore it doesn't interact with it's typically inert, and so it doesn't interact with any its surrounding media. And so people have been using nano diamonds inside cells, and in fact we have now started working towards looking into NV centers with nano diamonds. I'll show you some results, and you can see that NV centers have been used for measuring the concentration of biomolecules, and by tagging them with different fluorescent diamonds, you can use NV centers as a very low concentration detection of biomolecules. And then you also have NV centers which can image, for instance, a magnetotactic bacteria here. You can actually look at the magnetic profile of bacteria and be able to understand its ambient conditions. There are other applications, and one of the things that we have been very interested in is this C. elegans worm. C. elegans worm has one single neuron, and neuron is like a current carrying wire. So essentially what it does is that it emits all these axonal potential when it travels down this axon, and that it has been measured with a bulk NV center sample that is sitting underneath this neuron. And this really has been one of the motivating work that really enabled us to think about how we can not just image one single neuronal signal, but actually image the brain and understand how the axonal potentials are traveling. And if we can really able to understand the location of, or the origin of these the axonal potentials. And the greater goal is essentially to understand how neurons interact with each other, and if you have any kind of stimulation, how does the brain really perform when it comes to sensing or when it comes to sensing or perceiving any stimulation, and how does it behave? And that can only be done if we understand more of the neuronal behavior. And this has been the greater goal that has motivated me towards developing the magnetic field imager that we are interested in. Another application that I've always been interested in is understanding biological systems and how they perceive magnetism. And this is something that we've also been working on the side along with. But now I'll focus on really the neuronal segment, and developing a dynamic imager that will be able to image such sort of neuronal potentials. And so, of course, we are physicists, we develop tools. So it's a spherical cow-like approach, and what we have essentially identified is that each of these sort of neurons have this little wire-like structure. These are nothing but wires, and what we would like to do is understand the interaction of wires and the current flowing in wires, and see if we can actually map, first order, can we actually map the magnetic field from these kind of wires at all. And that is been one of the main primary goals with which we started. And so we started simulating something like a coil-like structure, which will have like a large head. And you may find some similarities, which is why I say mimic devices, is that it is a large head and a long tail. And so it's similar to an axon, a neuron, which has a large, a body, a cell body or a soma. And then you have a long tail of axon. And so essentially in this case, it's a long tail of the wire. And so it will have a large magnetic field, which will be at one location, and then we'll just have a flowing sort of path. And that's the mimic devices that we've been trying to image. And I'll tell you what our problem statement really looks like. We wanted to develop what is known as the sensor understanding if we can at all measure neurons, neuronal potentials in form of current flowing in wires, understand how we can reconstruct it. So it's not just magnetic field. Remember, magnetic field is a vector quantity, and when you have a sensor that is essentially in measuring a net magnetic field, so you have to be able to reconstruct back the actual location of the magnetic field and the amplitude of the magnetic field. So there is some sort of algorithm that is necessary to understand how you can extract these information. Right. And so then we want to develop something of a prototype, and hopefully one day we will be able to go towards single neuron imaging. Now, with that, we started developing models for neurons, and neurons are very well understood. There's a very nice, circuit model for neuron, which has been developed by Hodgkin and Huxley. And what you can essentially do is understand how to calculate the current that is flowing inside a neuron by modeling it, because with respect to the ion channels that are there, the sodium potassium ion channels that are created, that allows you to actually calculate what is the current that is flowing inside a neuron, and from the current that you can actually estimate back what is the B field. And now in this case, we did a much better 3D modeling of a neuron using what is called as a neuron model, and try to understand what are the features of a neuronal current. And interestingly, we found that the magnetic field is actually very highly localized inside the point which is known as an axon hillock. Now, axon hillock is right between the axon and the starting point of this, the cell body. And so this point has very high magnetic field, unlike the entire tail. So you actually don't need to measure the entire tail if you can really try and see that the magnetic field is pretty much localized in that place. So we started doing this in very detailed modeling, and if you're interested, you can find all the details in this communication physics paper. But what I would like to sort of highlight here is that when we did the modeling, we understood, we basically saw the two features that are very key, which is essentially when the neuron axonal potential travels, it actually splits and goes in two directions. And here in this, you can see that the yellow two blobs actually, tells you that the features are propagating in two directions. But an interesting feature that also emerges from the modeling is that it's not just a dipole-like feature that you would expect. It's actually a quadrupolar kind of behavior from the magnetic field. And the fields are of the order of some picoteslas. And so it's about 30 picotesla, and that gave us the confidence that indeed it may be possible to actually measure the magnetic field from a neuron and be able to extract out the neuronal potential from a cluster of neurons. And nobody has done this, and this is an open problem today. Then the interesting thing that we developed is a method or an algorithm which will allow us to extract the locations of the neuron, and that is using what is known as a matching pursuit algorithm. So we build a dictionary assuming that the neurons can be in a certain XYZ location of a voxel or a volume of material. And we estimate what the magnetic field is that will be measured by the NV center. Now you then use you basically use the NV center to measure a whole bunch of, signals that you would, which is essentially a vector sum of all the magnetic fields it would measure from different neurons that are firing. And then we do what is an inverse problem, and we solve for different, so we take this product and we do it in a sequential manner to identify the neurons, because a certain neuronal signal will always last for about a millisecond or so. So at least 3 of them should, subsequently, match, and we set a threshold for that. And we identify this sort of method of, doing the matching pursuit algorithm. We run it several times until we actually identify all the neurons. And the conclusion of this work essentially is that if you have a neuron or 2 neurons that are separated by at least 20 microns apart, you can distinguish them very nicely if they also temporarily fire at least 1 millisecond apart. And this we can do at least with 80% confidence, and we can really locate the entire volume of neuronal signals. And this was essentially a sort of a theoretical work that we did to understand how the neurons would behave if we were to use an NV center to measure the neuronal potential. Now, we moved on to then start working on developing the imager, because ultimately the imager has to be able to measure that level of magnetic field and be able to, understand how we can, develop this sort of a sensor. We started off with this diamond, which is essentially a layer of NV centers, and we then started building our setup, which would be like the NV center, which would be able to at least. So you have to be able to also track the, the magnetic field changes at the level of milliseconds, and nobody showed this. All sorts of images that were developed till we did this work, they were all static. It took about 2 to 3 hours to form an image that with high enough fidelity you can actually distinguish all the spatially resolved features in a magnetic field map. And so we were like, okay, what is the way out in which we can actually do a temporal imaging of the magnetic field features, and what would be an ideal way of doing this? So we started off with the standard things of calibrating a sensor. And so we did perform the ODMR. We started looking into the, the details of the ODMR. Here you can see 2 split at 3.1 megahertz, and this is because of the hyperfine interaction between the nitrogen nuclear spin and the electronic spin. So essentially the resonance can be modeled as D plus, all these terms because of the magnetic field as well as the electronic field. If you have a strong enough bias field, which we applied, you can actually work in the regime where the NV centers are only affected by the B field. And so that's what you see here. So if you apply a bias field, you can see 4 lines as one would expect from a bulk NV center sample. So when we performed, qubit control on this sample, so we performed Rabi, very nice looking Rabi. You can see that it almost goes up to 8 microseconds. We can control the spin state very precisely. And then we perform T2 star measurements, which is essentially coherence measurement and magnetic field sensitivity. And here we have a short noise limited sensitivity of about 150 picotesla per square root hertz. Now, you can then perform Hanh echo and all the different kind of ranges of, all the NMR has defined a whole bunch of, sequences that you can perform. We did some very simple ones like the Hanh echo here. And then we've also done XY8, etc. You can see that very nicely. You can see that the echo is extending the coherence time, or the T2 of these NV centers. Of course, here you can see some more modulation, and that's because of the nuclear spin interaction with the electronic spin, and that comes up like a double electron-electron resonance or nuclear spin resonance. Now, in between, we decided that to understand, this is a very I'll just take 2 minutes in this work, because, any sort of setup is fairly complicated, because you have microwave, you have optics, you have all sorts of things that are working in synchronization with each other to actually get this ODMR signal. Now, my student was trying to put together a smaller setup and seeing that are there methods by which we can actually avoid all sorts of things? And we came up with this strategy, which is known as the FFT technique, which essentially is that if you just modulate the microwave signal and you look at the output of the green light, the red light here, the red light is actually even and gets also modulated. And if you just take an FFT, you can essentially reconstruct back the ODMR here. So if you take the FFT and then take the maximum of that FFT at each point, you can really get back the signal. And the interesting part is here is you don't have to do any synchronization, you don't have to do any switching, nothing is needed. You can just take the optical light on the photo detector, take an FFT of the signal that you are performing, and then you can get back the signal very nicely, and you can get a sensitivity of 139 nanotesla. It's not too bad when if you want to do it for a teaching lab, especially, and we did this with the motivation of developing magnetometers for teaching labs and quantum teaching labs. And so here you can see that you can essentially create a very small, simple structure. So it's almost looks just these components are needed, and you can get back the ODMR. So this is something that we did on the side. Now I'll go back to our main work, which is frequency modulated technique for investigating and developing a wide field imager. So we perform frequency modulated optically detected modulation here. So instead of seeing a Lorentzian, which I showed you earlier, you actually see a derivative of the Lorentzian here. Here we are frequency modulating the microwave, and you can see the 8 peaks are very nicely separated here. And then we wanted to extend this work to the wide field, and for that we used a lock-in per pixel lock-in detector, and you can see that each pixel now has this ODMR signature. That means that if you have a wide area which has several pixels in it, each of the pixels have different ODMRs, and ODMRs that correspond to the spatial profile of the magnetic field that is affecting that particular location. And that's what we did. And so essentially, then we went on to understand the sensitivity per pixel. It turns out to be about 700 or so nanotesla per pixel. And it has some distribution, of course, because the NV centers are not uniformly distributed. You have some sort of a distribution of NV centers. So of course, the sensitivity is dependent on the nature of these NV centers in the sample. It's very sample dependent, of course. And then we built what is for understanding static magnetic field imaging. So as you can see here, we have a coil that we were imaging, and we were flowing the current through the coil, and then we placed it next to the diamond sample. And I'll just show you with the what if the setup actually looks like this, which is a very large setup here, and you have all sorts of electronics. And finally, when we wanted to perform the mimicked sort of experiment here, we were flowing the currents in these coils here, and you can see that I'll show you what the data really looks like. So if you did a static magnetic field imaging, this is the simulation. This is very easy to do in COMSOL. You can just simulate the magnetic field by just flowing the current in and then mapping from the Biot-Savart's law. You can calculate exactly the B field, the Bx, By, Bz, and then in the experiment, when you perform the experiment, you can see that the single NV axis can actually very nicely map out the Bx, By, Bz fields. Okay. And then we were like, okay, but this is all static. What happens when we flow temporal currents? And again, when we started to modulate the current in this wire, you can see very nicely the traces of the flowing, the current, modulated current can be easily tracked back. And in fact, if you take an FFT, you will also see the harmonics of the current that is flowing inside this wire. And here you can see the color contrast essentially tells you that the B field is rotating around the wire, as you would expect. Essentially, it's a vector field. So here it is one corner of the wire that has been imaged. And then we went on to look at the current flowing in in a coil, micro coil, which is essentially our mimic device. And here you can see as the phase of the current is flipped or it is modulated, you can see that the field also flips as per the color. You can see it's red and blue, minus and plus. And if you take the FFT, you see here we have a a 15 hertz signal. And so, and you can essentially see the harmonics again as well. Now we've done this and we've seen that this can essentially go up to almost 30 milliseconds or so, although we are not hitting the, the 1 millisecond target that we have yet, we are at least able to do 30 milliseconds. Now, of course, there are various other people have been also looking at this, and they're using very complicated methods of sort of developing this wide field magnetometer. But we have made a very simplified setup, and that is why we filed a patent for this. Now, one thing that we would like to show here is that when we are interested in looking at, neuronal signals. So let's say that we modulated a current that is not a periodic signal, for instance, here. You can see that you can actually track between these two windows. You can track that at the point where it's changing sign, there is no magnetic field signature. So it means that at least at the 30 millisecond time scale, we are able to track magnetic field changes, and this is what we have a patent for. I'll just show you one quick video. So here, essentially what you have is a multi-coil like structure, and as you change, flow different kind of current with different phases, you have different magnetic field, and that is created. And this is at a video frame rate. You can see that the magnetic field can be easily tracked, and this is now what we are going to use to develop the magnetic field sensor. Now, a quick aside, and in 5 minutes, I'll just tell you. So we have been now interested in applying this technique, not just to current carrying coils, but also to understand, can we do other measurements like magnetic field AC susceptibility measurements. And so for that, we started working with these nanomagnets, which are essentially patterned on silicon dioxide. And here you can see very nicely the dipole. So this is an experimentally measured magnetic field map of a nanomagnet that has been patterned on on these on silicon. And then this is the best fit simulation. You can see a very nice dipole pattern that has been measured by, this is a measurement of with NV centers. Now, what we wanted to understand was that, can we actually do AC susceptibility measurements? And for that, we developed a new technique wherein which we control the magnetic field and similarly sense the magnetic field with the NV centers. And from there, we extract the AC susceptibility. So we can do an on-off measurement and compare and understand the real and imaginary parts of the magnetization using these NV centers. And if you're interested, this paper has come out in Reviews of Scientific Instruments. You can read about the technique that we developed. Now, I want to quickly point out one more thing, which is talking about cells and magnetic biosensing with NV centers. And this is work we started about 4 months or so ago. And here you can see this is an image of a stem cell. This is a human mesenchymal stem cell. So what we were doing here is that we were trying to understand whether the cells will at all engulf the nanodiamonds or not. And so here you can see that this image basically shows the cell. But to identify, biologists know that to identify cells, you have to do something what is known as a DAPI imaging, which is essentially to map the green fluorescence that is emitted by nucleus. So you stain the nucleus and then you map the fluorescence image of the nucleus. So here you can see a confocal image with the nucleus, which is identified very nicely with this stained technique. And you can see some black dots are inside this. Now, when we performed our standard fluorescence microscopy for NV centers, which is essentially to look at the NV fluorescence, you can see the red dots are here, and it tells you that the cells have really engulfed this the nanodiamonds. And then we performed even some more experiments, and in fact, we have now also done Z-stack imaging, where you actually identify whether the nanodiamond is sitting on the top of the cell or it's actually inside the cell, or underneath the cell. All of those image measurements we have now performed, and we are trying to now get into the regime where we can do localized magnetic field sensing with these cells, or even any kind of biomolecular sensing inside the cells. And that's something that we are now moving towards. Another thing that we have done in the meantime is essentially what is known as a relaxometry measurement with nanodiamonds, where in the presence or absence of magnetic nanoparticles, the relaxometry parameters of T1 actually gets modified. And the T1 measurement is very simple. You don't need any microwave. All you do is you just map the fluorescence of the NV center and keep mapping it over time. And as you can see here, so it's essentially the decay rate of the the eigenstate of, so it's with NV centers in the ground state 0 or the plus or minus 1. You just look at the decay of the fluorescence. And here you can see that in the presence or absence of the T1 gets considerably modified, and even in the ODMR that shows up. And so we've been looking at the photon statistics and the ODMR, and in the presence or absence of magnetic nanoparticles are easily detectable. And now we are trying to push it towards, understanding how, we can get it to really low concentration detection of magnetic nanoparticles. Now, with that, I will summarize what we have been doing so far. We've been working on developing NV centers for magnetic field microscopy and various different applications: temporal magnetic field imaging, micromagnetic imaging, and cellular imaging, and moving towards different kind of applications of NV centers. So with that, I'll thank you very much for your attention. And also I have to thank the funding agencies. So DST Quest program has funded us for all the single photon work, and also the single qubit work that we have been doing, and also the sensing work partly that we have been working on. And IIT Bombay gave a generous seed grant, and we've been working with that. SERB, Inspire, and US Air Force has been funding us and also funding us now for looking at temporal magnetic field imaging. And this is my group, and there are some changes now, more people added, and many people are now graduated. Madhu has now graduated, and there are several collaborators that I also have to thank. And with that, I'll thank you, and I can take some questions. So I'll just stop sharing and I can take some questions. Hello?
The floor is open for question. Anyone has any question? Okay, I have a question that so how the can you use it to measure the gravitational potential or something?
Gravitational potential. See, NV centers have been shown that you can actually do inertial rotation sensing, but not really gravitational potential yet. I don't think the sensitivity is enough to actually get to that regime. Yeah.
Okay. So if anybody, no, I don't see any other question. Sorry.
Hello, can you hear me?
Hello?
Hello, can you hear me?
Yeah, I can hear you. Yeah, please go on.
Yeah. Actually, I didn't attend the full talk, but I just now get to know that you are working in the NV Center and Diamonds. So one question I have relevant to this, that did you get any EPR or FMR spectra in these magnetic nanoparticles using?
No, we haven't done any EPR or FMR's work yet. We have been primarily looking at cells and trying to see if the NV centers work inside the cells or not. So we have not really looked into the, yeah.
Okay then.
So anybody else has any question?
Yes, I have one clarification question. Thank you for your talk. When you were talking about sensing, do you mean detection or what?
Detection, yes. We are talking about detecting very small magnetic fields and imaging them.
Okay. So when you were talking about sensing, it means that the state which you were trying to detect, it will be destroyed, and you are creating again once more the copy of that state and again doing sensing, right? It is a repetition process.
Yes, we have to repeat the process, but given the sensitivity, we can actually we can do single shot measurements as well. Yes.
Okay, but with single shot, you will destroy, right, the initial state, and then you have to.
Yeah, so we just look at the perturbation of the ODMR. In fact, we are not even doing any Ramsey where we are destroying any state or anything. We are not even, what we just do is essentially we look at the movement, we look at the shift of the ODMR. Okay, so here we are not even preparing the state per se. So we just look at, the shift of the resonance about a certain point. Yeah. So we are not doing any Ramsey or anything yet.
Mm-hmm. Okay, yeah. Thank you.
Yeah. Professor Usha, are you, so with sensing, I think there's a question. Yeah. Are you able to achieve Heisenberg limited? No, we are not. Actually, nobody in the world has been able to get Heisenberg limited sensitivity with NV centers. We are still in very much in the just the qubit operations. But we are looking into cavity interactions with NV centers to get to that. So some interaction.
So it is still within the kind of root n limit that you are working with.
Yes, root square. Yes, square root n.
Yeah, but still there is some kind of a good sensing with even within that limit.
Definitely.
Okay. Okay, thank you. And also I have a curiosity again. That is in avian compass kind of systems. What kind of magnetic fields, that is the range of magnetic field sensing, does that match with what you are looking at? That is the delta B is what I'm talking.
Okay, so avian magnetometry actually, so avian, it's like Earth's magnetic field. That's 50 micro. Yeah. That's very large actually, in fact.
Okay.
Yeah, it's very large.
So you don't need NV centers to kind of simulate it.
No, we don't. Yeah. Those are very classical sensors. Yeah.
Thank you.
Okay. Any other questions?
So any other question, anybody? Not in chat or so. Other questions? Let's thank the speaker. Thank you, Dr. Kasturi Saha.
Thank you very much. Thank you. Yeah.